Dr. Perkins, and this is my first time at BIF, and I have to say it's a great meeting, and it's a pleasure to be here. With a clarification that I am a feedlot nutritionist, and so I'm going to attempt to turn a genetics talk into a nutrition talk so that it's something that I understand and feel comfortable talking with you folks about. Also, I'm going to keep it at a very high level so that I understand it to present it to you, and then so that Dr. Perkins can understand it also. So with that, we'll go ahead and jump right in. So today I'm going to be talking the first half of my talk about transcriptomics. And so this sounds like a big scary word, but it is really just how DNA gets expressed. So genes get turned on in a process called transcription, and then RNA that gets made during that process are called transcripts. And so the entire collection of RNA is called the transcriptome. And so this is ultimately the information in the body that gets turned into a protein. And so you may be thinking, well, why is this important, and why in beef cattle do I care what a transcriptome is? And so we need to study this because we need to understand what kind of polymorphisms are contained within coding regions of genes on the genome. And so within these coding regions, we're really looking for functional variants. And so a functional variant is something that translates to a phenotypic change. So it's something that you as producers and ranchers can actually see, versus sometimes we have variants that don't actually code for a protein, and so you can't tell that they're there. And so this is important in our genetic selections because we need to know which markers are most important in our genetic evaluations. And so you heard a lot about that in the main sessions yesterday and today. So we're going to start by talking about why do transcripts vary. So what can cause transcripts to vary in cattle? And so they vary in response to external stimuli. This can be things like their environment. I imagine if we took a cow from Lubbock, Texas and dropped her into Las Cruces, New Mexico, that causes some change because it's a very different environment. Stress can cause these to change, nutrition, whether that be under or over nutrition, and then other external influences like animal husbandry. And so these transcripts, other things can affect that, such as disease. A calf has different transcripts than a mature cow, so their stage of growth and stage in life can also impact why their transcripts vary. And so I've got what I'm calling a tale of two calves here to try and illustrate how the transcripts can vary and how environment can influence that. And so if we have two heifer calves that were born near Canyon, Texas, they would have a very common environment in early life. And then let's say hypothetically that maybe around a year of age they got sold. One of them stayed near Canyon, Texas, and one went to Missouri. And you can tell in the picture which one is Canyon because there's a blade of grass about every 2 feet this time of year, and then which one went to Missouri to the knee-high lush green grass, and those cows have seen rain for the first time. But these transcripts can affect things like milk production. They can affect their energy requirements, and so as researchers we need to understand the molecular basis for these change in transcripts and how that affects the phenotype. So how it affects you as ranchers and producers on the bottom line. And so that's why we do all this fancy genetic work, is because we want to know how that impacts your bottom line. So the first study I'm going to talk about, we looked at differential transcript abundance in adipose tissue of mature beef cows. So staying along the whole body tissue theme, we're going to be talking about adipose tissue. And so I must have made this slide back when the price of feed was cheaper, because I say it accounts for greater than 70% of the annual expenditures, but we all know that that number can be maybe 90% today. So feed is the biggest cost of these operations. And so cows in your operations, they use the feed to support conceptus growth, like Dr. Scholler-Gerdes talked about. They use it for milk production. They use it to provide energy so that they can graze and do locomotion, and that becomes especially important in extreme environments here like Las Cruces, where I imagine that they are walking further to graze and do locomotion. And then ultimately, they use that energy to maintain their body weight. So beef cows often experience periods of nutrient restriction, just like Dr. Scholler-Gerdes talked about. And this can be something that's not man-made. It can be restriction because of gestation. Maybe the forage is not good enough to support third trimester gestation. It can be because of lactation, or just flat out that there's a drought and forage quality is low and there's not much of it. And so cows respond phenotypically by changes that may be occurring metabolically at the tissue level. And so this means that something you can see as ranchers happens because of something that's controlled by the metabolic tissue level. And so here's where I attempt to make this a nutrition talk. So one of the things that could be happening during times of restriction in your cows could be some compensatory growth. And so if you're not familiar, compensatory growth is a time of restriction. So it's a time where there's not enough for the cows to maintain their body weight, and this applies to growing and finishing animals as well. They decrease their fat reserves to maintain their body weight. Their metabolic rate, or their heat production, decreases. They also decrease lipid deposition because they're mobilizing those lipids to give themselves energy. And then also their visceral organ sizes decrease, so their gastrointestinal tract and their liver. And so that's what drives down their heat production. And then whenever forage is plenty and they come out of the restriction in the recovery period, they are very feed efficient because they decrease their metabolism. They have an increased lipid deposition. Their feed intake is usually increased, and their visceral organ size comes up. And so these shifts in metabolic rate suggest that cellular and tissue responses respond to energy availability. And so we really need to understand these molecular mechanisms behind this process, because we've observed compensatory gain in the beef industry for decades now, but on a molecular level, we do not understand what controls that. So that brings me to the first study that I'm going to talk about, is do cows that differ in efficiency of weight gain differ in the abundance of transcripts that regulate adipose tissue. So this is a talk that's all about their adipose and their fat tissue. So we used mature cows that we subjected to a feed restriction. Our restriction was for 112 days. We restricted these cattle to 0.7% of maintenance. So they were losing weight every day. This was not a welfare issue or dangerous, but they were losing weight every day, but was not outside the realm of what would happen during times of drought. We restricted them for 112 days so that they were adapted to the restriction, so that their bodies were not rapidly changing when we collected these samples. And then after 112 days, we gave them back the same diet that they were restricted on, which was the high forage diet, so that it translated to ranchers and cow producers like yourself. So we restricted them on a high forage diet, and then we gave them all of the diet that they wanted back ad libitum. And so you can see that these cows really split themselves into cows that did really well and were high gainers during the ad libitum refeed period, or that were low gainers and didn't gain quite as fast during that ad libitum refeeding. And so we collected adipose tissue biopsies on day 105 so that they were well adapted to that restriction. And then we collected biopsies again 49 days after we started the refeed, so that theoretically they were well adapted to the refeed. And so we used open, dry, mature cows. And so we did this on 100 cows, and it was kind of a feat to get the center director at Meat Animal Research to say that we could have 100 open cows for this study. But we didn't want anything else going on. We didn't want them growing a calf. We didn't want them lactating or anything else. And so while we did the study on 100 head, we picked 6 cows that were our highest gainers, and then 6 cows that were the lowest gainers during the refeed period. And so from those adipose samples that we took, RNA was extracted and microarray was used to determine differential expression of genes in the adipose tissue of these mature cows. And so our results, what we found was if you looked only during feed restriction, we found 113 genes that were differentially expressed in adipose tissue between the high and low gain cows. So now some of these genes were upregulated and some of them were downregulated, and it's not important to know how many of each, but many of them were involved in lipid metabolism, which is something that makes biological sense to me. If I'm taking feed away from you and you're below maintenance, the genes that you're expressing are involved in how you process lipids. Then during the ad libitum refeed, again, we found about the same number of genes, about half as many genes that were differentially expressed between high and low gain. Some were upregulated, some were downregulated. We didn't have any pathways that were just really interesting during the ad libitum refeed. So then we looked at the data a different way. So we only compared high gain cows to each other. So we're only looking in the high gain cows, but now we're looking at the time that they were restricted versus the time that they were refed. Well, now we found roughly 500 genes that were identified. Most of these were upregulated, only a few were downregulated. So most of these genes, interestingly, were involved in propionate production, and propionate is a volatile fatty acid in the rumen. It's how ruminants absorb energy, and so the feed that they eat gets converted into a volatile fatty acid, and then they absorb that and use that for energy. And so, you know, what strikes me is the high gaining group, what distinguished the high gainers in the refeed was their ability to produce propionate. And so something about them, they were just a little more efficient. But then as we look at the low gain cows during the restriction versus ad libitum refeed, we found roughly 500 genes again. Most of them were upregulated, some were downregulated. Most of these genes were involved in things like cholesterol biosynthesis, which is a precursor to steroid hormones. And so those cows were just worried about trying to have hormones to reproduce, etc., to maintain homeostasis within the body. And so that is something unique that we found in those low gain cows. And then so just to kind of summarize this study, you know, what made the high gain cows unique was that they used their energy to focus on energy production within the body and reproduction. And then the low gain cows, the genes that they differentially expressed, those were mostly used in cholesterol biosynthesis, which is ultimately used for steroid hormone production within the body. And so what does all this mean? So, you know, my boss used to always tell me, well, so what, who cares? You know, how can you explain this? So this is my so what, who cares slide. So the expression of energy production genes were lower during feed restriction than refeed, and that makes biological sense. If you don't have enough feed to eat, you're not concerned with energy production. The specific transcripts responsible for these changes differ between animals that respond at a high or a low rate during the refeed. And then these pathways critical during refeed included energy production pathways. And so I think this is just a really interesting study to where we can have some very divergent phenotypes, and then we can look at something that is a known phenomena in the cattle industry and then tie it to a molecular mechanism. And so, you know, that was our approach. This is not a lot of numbers that we used, but we found very big differences. And so we considered this study a success. And so now I'm going to pivot a little bit, and instead of talking about gene expression, we're going to talk about marker-assisted management, or what you may have heard as marker-assisted selection. And so it's important for us to investigate the effects of SNPs on genotypic and phenotypic changes. And so at the Meat Animal Research Center, we had a unique composite of cattle that was called the Mark I composite. And so it's 1/4 Limousin, 1/4 Bromby, 1/4 Charolais, and then 1/4 Angus by Hereford. And then the allele, the F94L, for the myostatin gene was selected for in this breed, in this composite breed, because again, it does include Limousin cattle. And so the F94L myostatin mutation is prevalent in Limousin cattle, and in purebred Limousins, I think this can be, anywhere between 80 and 90%, so it can be quite high in purebred Limousins. And so it increases muscling and decreases fat. So this is more of a moderate or modest phenotype change. It's not double muscling like Belgian Blue or Piedmontese, but you can see a distinct difference in protein accretion in these cattle. And so it's important to study because effects on growth, maintenance energy requirements, and carcass characteristics, and there are enough Limousin cattle fed in this country that it's important that we have, research and data on this. And then my interest as a nutritionist are on the maintenance energy requirements, because what that means to you is how much forage does that cow need to maintain her body weight. And as Dr. Scholigert has pointed out, there can be a little stress in there. And so ultimately, how much forage mass does she need to maintain her body weight? And so I do think a picture is worth 1,000 words. And so I borrowed these pictures. I couldn't remember if they were from Gary Bennett or Larry Cundiff, so I just thanked them both here. But these are actually pictures of the Mark I composites from the Meat Animal Research Center. So on the top, these cattle have 2 copies of the F94L mutation. The middle, they have 1 copy, and then the cattle in the bottom, they don't have any copies, so these are the homozygous cattle without the mutation. And so when I look at this, and I looked at a fair number of these cattle over the years, I think I can distinctly see muscling differences here in the hindquarter, over the rump. They usually had a thicker shoulder, and so I think I could see that phenotypically, certainly. When I look at the cattle in the middle, I don't know if I don't actually see something or if it's because I know they only have 1 copy, but definitely you could look at cattle without the mutation and they just look like average cattle. And so, I created a muscle scoring system and scored maybe 1,000 of these over the years, I don't know, just for fun, and then never did anything with it. But when the cattle with 2 copies left the chute, they looked like a little meat wagon running down the alley. And so you definitely could tell that they had 2 copies. And so my study has nothing to do with steers, but I just wanted to show you some pictures so you knew kind of what we were looking at here. So the study I'm going to talk to you about, the last study, is about growing replacement heifers that were from the Mark I composite breed. So now we genotyped some heifers and we selected 12 with 0 copies of the F94L mutation, and then we selected 12 with 2 copies of the F94 mutation. Now we didn't want any heterozygotes in the study, we just wanted to look at the 2 extremes. And so our whole goal was to say, if there's a difference in muscling and a difference in protein accretion, does that change maintenance energy requirements? Because I had the interest as a nutritionist. And so to get at this, we had to use these head box calorimeters. And so it's where you put their head in a box. You can measure how much oxygen they consume, how much carbon dioxide and methane they produce in a 24-hour period. And then you can plug those numbers into an equation and estimate how much heat they lost. And ultimately, losing heat is the same thing as their metabolic rate in cattle, or their fasting heat reduction. And so we wanted to really get at this maintenance requirement, was how much energy did it take to maintain their body weight, and did that differ whether they had 2 copies or no copies of the F94L mutation. And so what we found is that heifers with 2 copies of the F94L mutation consume less oxygen, and that's important because oxygen can be a proxy for metabolic rate. And so I'll be honest, I was a little surprised about that because I thought, man, they have more muscle, but yet their metabolic rate's lower. So it kind of got my wheels to turning. Heifers with 2 copies also, when we did the equation, actually had less heat production. So what that means for you is that there's less forage wasted. You know, you can maybe stock a few more animal units if you have something with less heat production. And so again, I was perplexed how a more muscular animal could have lower heat production because muscle, you know, is tied to protein turnover, and protein constantly turns over in the body. And so I thought, well, it couldn't be that they had more synthesis to have more muscle because that would increase the energy that they needed. So it must be that the myostatin decreases protein degradation, so it just turns over more slowly. And so that's how they have a lower heat reduction and decreased metabolic rate. Although we didn't measure any of that in the study, but that was just what we concluded. And then lastly, for body weight gain, heifers with 2 copies tended to gain weight faster and maintain weight easier. So these heifers with the F94L mutation, 2 copies, I would term to be an easier keeper. And this is again likely because of their decreased metabolic rate. The other interesting thing I ran across in the literature, although we did not measure, was that these heifers usually gained maturity quicker, probably just because maybe they gained weight a little bit faster and reached that mature weight or that puberty weight a little sooner. But there is some data to indicate that they're a little bit early maturing as well. And so, you know, to kind of tie it all up in the big picture, so, you know, what's the big picture here? What's the take-home message? So what genes are associated with biological function? And when I think of this, I think of on the same forage resources, like the cows in the picture, why do these Charolais cows differ in their ability to maintain body weight or to grow different sized calves, even if these cows are highly related? And could it be that they express their genes differently? So, you know, future applications, this is not just for the studies we did, but for genomic selection in general. You know, we need to continue to use this genomic technology to greatly increase our rate of genetic gain while it's available. We can also use these accelerated gains to meet our future demands for food because our population is growing. There's plenty of talks about that. And then specific things for genomic selection are we can select cattle that are more capable to handle these environmental stressors that I discussed, more capable to handle heat tolerance, more resistant to disease, have increased meat quality. Now that could be something like a decreased proportion of dark cutters in the same pen. We can also use genomics for meat safety. Maybe we find that certain cattle are predisposed to shed E. coli versus others are not. And so there's a lot of applications here. Increased feed efficiency, and then of course, always increased reproductive efficiency. And so this slide is just more of the same, you know, but we certainly can use genomics to study bovine respiratory disease or shipping fever. As a nutritionist, I'm interested in liver abscesses and what causes those and what we can do to prevent those. So using genomics for that, and then also these cattle on the end don't have pneumonia. This was just a hot day in Clay Center, Nebraska. And so, you know, selecting cattle for the right environment. And ultimately, we want to increase the utility of our genetic evaluations. And that's all I have.